Alright, you guys, I am going to show you some variations on Mendel's theme. The first one is called incomplete dominance. And this is when you cross two purebred organisms and the phenotype of the F1 appears completely different than either of the parents. And it's an intermediate phenotype between two homozygous parents. This happens a lot in flowers, so I'm going to use flowers as my example. The flowers I'm showing you are called snapdragons. If you were to cross a homozygous red with a homozygous white flower, the result is all pink flowers. Show this on a Punnett square. You have a homozygous red, which is big R, crossed with a little r, that would be the white snapdragon. The genotype that you get is all heterozygous, but the phenotype that you see would be all pink flowers. What would happen though if you crossed an F1 pink with an F1 pink? Now this is where when Mendel had done his crosses, remember we did purple flowers and white flowers, he wanted to see if it was blending, and he did not get light purple flowers or violet flowers. All of the flowers came out purple like the parent. But this also demonstrates that it's not blending, because let's see what happens if you cross two of those pinks. So the big R, little r is the genotype for each one. What you see, you get this genotype ratio, 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive. But the phenotypes are interesting here because you get 1 red, that's your homozygous dominant, to 2 pink, the heterozygous, and 1 white flower, the homozygous recessive. So again, it wasn't blending. Otherwise, how would you get the white and the red to come back? And think about what it would be like if you mixed red paint and white paint together and got pink paint. There's no way you're going to be able to get that red paint separated back out. So what would happen if you crossed a pink with a white? So pause the recording for a second and draw a Punnett square of that and tell me what you would get for your genotype and phenotypic ratio. So setting up a Punnett square should look like this. You have your pink flower here and the alleles for the white flower here, and you should have gotten 1-to-1 genotypic ratio, heterozygous to homozygous recessive, and your phenotypes are also 1-to-1, or you could say 50% of them are pink and 50% of the flowers would be white. Type this link into your browser, and there's questions there that you can practice at the end of this. Questions 1 through 3 are really good. They have multiple parts. You can ignore questions 4 and 5. And we're going to practice more of these tomorrow in class. So try to see if you can do those questions 1 through 3 on your own. And if you're having trouble, we're going to do them in class, and I can help you there. All right, here's another variation on Mendel's theme. It's called codominance. And this occurs when there are more than 2 forms of an allele. And 2 of these forms can be equally dominant. Now, so far we've been talking about forms of an allele when there's 2 forms, a recessive form and a dominant form. However, there can be more than 2 forms. Human blood type is a great example because there are 3 forms of this allele. There's the A form, the B form, and the O form. Okay? Turns out that A and B are equally dominant to each other, and they're both dominant to O. So when you express these alleles together, it's often common to use the letter I and then the subscript. So this individual would have one allele that's A, so you say I subscript A, and one allele that's B, so it's I subscript B. I tend to not draw the I's, and it's more straightforward if I just draw the letters. So here's the thing. When you look at somebody who has blood type A, and they know their blood type A, there's no way to know if they have 2 A alleles for their genotype or an A and an O. So when a person is type A, they're either homozygous for A or heterozygous for A and O, but their phenotype is type A. The same thing for someone whose phenotype is type B. They have 2 alleles, and you don't know if they have 2 B's or if they have a B and an O for their genotype. A person that is blood type AB, their genotype is one A allele and one B allele. And a person that is blood type O has a genotype that's two O's, because O is recessive to A and B. If they had either A or B, that would be their blood type. If you did a cross between a person with type A blood and a person with type B blood, could they have a child with type O blood? Okay, so why don't you pause the recording and try that? So the answer would be, if you don't know their second allele, they would have a possibility of having a child of AB, type AB, but they could also have a possibility of having a child with type B blood, a child with type O, or they could have a 1 in 4 chance of having a child with type O blood. Without knowing what their second allele is, there's no way to know. But if they have an offspring that is type O blood, then you know what their second allele is. They had to have been heterozygous. So the answer is, it is possible that two people with type A blood and type B blood could have a type O child. They could also have type AB, type A, type B. What blood types are possible if a person with type AB blood and a person with type O blood mate? Again, set up a Punnett square and try this yourself. If you set up a Punnett square and it looked like this, where the person with type AB was at the top and the person with type O, it doesn't matter if they're on the top or the side really, but here's the type AB person and here's the type O person. They have a 50% chance of having a child with type A blood or a 50% chance of having a child with type B blood. Again, here's a website that has some of these problems to practice. Actually, this website has a bunch of different problems. All of this point that there's monohybrid crosses and dihybrid crosses, codominant and incomplete dominant. So there's plenty of there to use for practice. The third type of variation I want to point out is called X-linked genes. Now, so far we've been talking about genes that we would expect to find on autosomes. And remember, autosomes are chromosomes that are not sex chromosomes. So anything for an organism that doesn't involve sex determination. And for humans, that's the X and the Y chromosome. So you can understand why if a disorder is on an X chromosome, it's going to affect humans differently because females have 2 X chromosomes, while males only have 1 X chromosome. So my example is red-green color blindness. It is an X-linked recessive disorder. Now, recessive means that it will not matter, the recessive will not show up if there is a dominant allele present. So if you have a female, she has 2 X chromosomes, and we'll use the R, this little subscript R, is their red-green color blindness gene. So the color blindness gene. So this person has 2 large R's, and she's homozygous dominant. This female is not colorblind. A male only has 1 X chromosome. This gene is not found on the Y chromosome. But a male with 1 gene, this large R, the dominant gene, is also not colorblind. A female that has 2 R's and she's heterozygous. So on this X chromosome, she has the dominant R. On this X chromosome, she has the recessive R. She is also not colorblind, but we would call her a carrier of the mutated gene. If you looked at a mating between this woman that's a carrier, so here she is on top, there's 2 X chromosomes, a big R and a little r. And over here we have the male who only has 1 X chromosome and he's not colorblind. So he has a big R. And you look at a cross. Now these two people have a 50% chance of having a daughter, the top row, and a 50% chance of having a son. Now if you look here, this daughter is not colorblind and this daughter is not colorblind. However, she is a carrier, but there would be no way to know that at this point. If you look at their sons, this son is not colorblind. However, this son is colorblind. So a cross like this would result in 0 daughters being colorblind, and 50% of their sons could be colorblind. They would inherit this mutant allele from their mother, and that would cause them to be colorblind. The Y chromosome is why they're male, and that's what their father gave to them. So typically, when a disorder is X-linked recessive, it is inherited and passed from the mother to the son. I will have more examples of these sex-linked disorders tomorrow, and we'll also look at an X-linked dominant example, although we'll focus mostly on this type of X-linked recessive disorder. So go through some of these a little bit slower if you need to, but we're going to have plenty of practice problems tomorrow in class. All right, I'll see you tomorrow.